All right, so thank you all for being here, and thank you for those of you who are streaming. It's my pleasure to be here to tell you guys about some of the work that my lab has been doing for the past 7 years. That has been really focused on nursery production of the native saltmarsh grass, Spartina alterniflora. And our purpose, as you'll see, we'll talk about, is to include and support the salt restoration of our salt marshes. I'll just start off by saying, how lucky are we to live in Georgia? Right? I mean, our coastal salt marshes are so expansive and so amazing. We have like 364,000 acres of salt marsh, which is pretty fantastic. And that salt marsh has become, I've only been here for 13 years. And let me tell you, already the salt marsh has become such an important part of my life, my day-to-day life. And so it's part of our culture as Georgians. I hope I can say that. I hope I've been here long enough. If not, I'm sorry. And they're also really important ecosystems in general. So they tend to fringe along the shoreline. So you can see on the map along the shoreline here, all along the East Coast and up along the Gulf Coast. This is where we find our salt marshes. They're extremely productive habitats. They're ecologically significant, so they support a nursery habitat for shellfish and fish, including things that we consume, so they have some economical importance, so it supports commercial fisheries. They buffer the shoreline from flooding, from extreme storms. Salt marshes actually filter water and reduce contaminants and pollution. I could go on and on. They support marine resources because they're exporting nutrients out to the ocean. So they're extremely important. So they provide a lot of ecosystem services. Unfortunately, most of the globally, salt marshes are in decline. And there's a myriad of factors. It includes habitat degradation through pollution, of course, land use change through urban development. All of these things can cause a loss of salt marshes. And they're also threatened in our scenarios of future global climate change. Along the coastline, we're expecting to have sea level rise, right? It's kind of a given now that sea level rise is going to happen. Just depends on how bad, right? And you can see that a lot of these salt marshes lay along these low-lying coastlines that are going to experience significant sea level rise. And so it's really important that we start to kind of think of a way to kind of help prepare our coastal system for these changes. So a little bit more about coastal salt marshes. Along the east and Gulf Coast of the U.S., the dominant salt marsh plant species is Spartina alterniflora. This is also known as smooth cordgrass. It is pretty much exists as a monoculture. So when you guys look out in the marsh and you see all that like awesome greeniness, right? That's all Spartina. And so because it is such a dominant species, it does play a vital role in the salt marsh ecosystems as well. Before I tell you about these ecosystem services, I will also note that Spartina is really, really good at tolerating a stressful environment, which is the coastal salt marsh. Spartina is able to grow in salty soils, and it is able to grow in soils with very little oxygen, and most plants can't do this. And that's one of the reasons that it is so dominant. It is important as contributing to high primary productivity because it is a dominant plant species, but it actually plays an important role in salt marsh growth and maintenance. It plays an important role in what we call the vertical growth of the marsh, so increase in elevation, as well as the horizontal growth of the marsh, which is an increase in the spatial extent. The above ground growth of the grass, the above ground growth of the grass slows down waves as they're coming in, and this helps sediments drop out and start to gather on the surface. And then you can see kind of this growth form where it has these really dense roots and it grows with stems coming out like this. That growth form below ground helps to stabilize and trap those sediments. So it's a very important plant in building marsh and maintaining shorelines. In addition, it is basically forms the base of the food webs. So anyone who likes seafood and shellfish will appreciate cordgrass because it not only is going to support our green or herbivore driven food chain, which is like things with snails eating them, but also the things we like to eat, like crabs, right? Those are supported also by Spartina because it decomposes, like crabs and scallops and clams are all eating that organic material. So it's pretty critical to our marshes. So all in all, I hope you guys appreciate that. Not only is the marsh part of our culture, but it's also extremely important to our coastal ecosystem. So to avoid losing marshes to this sea level rise that we're anticipating, the marshes have to increase in elevation at the same pace that sea level rise is increasing. And we can help do this by kind of doing these nature-based solutions to restore marshes. Here in Georgia, the DNR really is facilitating and kind of pushing this living shorelines, which is a great technique that uses green infrastructure like shelled oyster bags or coco coir and native biota to sustain and stabilize shorelines. These living shorelines have been found to be very effective in maintaining shorelines through hurricanes. They've also been found to basically have the same services as a native marsh. So they're restoring all those ecosystem services. They're providing habitat. And they've also been found to sequester carbon at a similar rate to natural marshes given time. Because of its importance in the important role in salt marsh maintenance and salt marsh ecosystem processes, Spartina alterniflora is commonly used and planted in these living shoreline projects. Okay. So Spartina has a really huge distribution, right? So it's found from New England all the way down the East Coast and around to the Gulf Coast as well. And there's been some research that has looked into the genetics to see how similar these populations are to each other. And what they've basically found is there's according to this one paper, there's four distinct groups, four distinct populations that exist along its distribution. And these different populations, these genetic differences are translated into differences in morphology. So like the plants in the south grow taller. They grow further apart, right? They put less below ground than the plants up north. And so they've actually grown these in common gardens and found like these differences in morphology are genetically, they're genetically coded, right? So even in common gardens, they keep those characteristics. They also have found differences in reproduction. So their flowering times are different, right? So they're flowering at different times. And this is a really like confusing kind of thing here, but what this is basically saying is these differences in morphology and reproduction translate into differences in this services that they give to the other organisms in the habitat, right? So the greater below ground biomass in the north is important for maintaining the other organisms and the other animals within that system. And so if you put a southern plant up there, the biomass is not enough to provide those services, right? So it's really important that we use plants that are within this populations, right? They've also found there's a lot of genetic variation within populations. And this basically suggests that sexual reproduction is important in maintaining Spartina populations, right? So they are producing seeds and they are producing seedlings, and that's contributing a lot to the maintenance of that population. They've also found that even within these populations that are distinct, there's variation within those populations, right? Especially in the South Atlantic. And they found that Georgia plants are really different from a lot of everything else, right? So combining all this research from along the distribution, studies are suggesting that when we put Spartina in these restoration projects, we use locally sourced plants, right? So they should come from within 100 to 300 kilometers from your restoration site. And they should produce by seed, because if you produce plants by seed, you end up with a lot of plants that are genetically different, right? And that allows them to be better able to adapt to their new environment that they're transplanted in and have higher survival, right? However, I will note that there is no nursery at this time in Georgia that produces Spartina, right? So it's a real need for these living shoreline projects. Traditionally, a lot of nursery production includes clonal propagation. That just means that you're like going out in the field, and you're taking, I use the root slayer. I don't know if you guys are familiar with that, but I use the root slayer. But basically you cut on either side of so these are all genetically identical. They're coming along off the same rhizome. And so they're all like, they are exactly the same, right? So we call them clones. And so if you take my root slayer and you cut on both sides and pull this one out and take it into your nursery and then propagate it, then all of those new individuals that you're growing are genetically identical, right? And so this traditional nursery methods means that you end up with a relatively low genetic variation in your stock, right? And you also, clones are grown in sub-irrigated trays, and they're fertilized with commercial fertilizers, which really isn't what they're experiencing in the marsh, right? So the purpose of my research and my lab's research has been to try to facilitate nursery production of Spartina by our native plant nurseries here in Georgia by providing information on basic reproductive ecology, like what are they doing out in the field, right? And also to try to develop propagation protocols, which I put some out here just to show you what we've come up with so far. I will say that it is not legal for you to go out and collect seeds in Spartina in the marsh, okay? Let me go ahead and say that right there. But we try to just get, brought up for you guys to see, but we're trying to develop protocols that we can share with nurseries to help facilitate getting this started. And so, oopsie. So, we've done a lot of stuff. Like I probably could spend about a mini semester, which is about 8 weeks, right? 7 to 8 weeks, telling you about our research in our lab. But I'm kind of focusing on our nursery production. And so over the past 7 years, we focused on looking at spatial and annual patterns in reproduction in coastal Georgia. This is just to better understand what our plants are doing in natural marshes. We've done experiments to optimize storage and germination conditions for seed propagation. And we've done that alternative methods to traditional nursery production. And those are parts of us trying to develop this propagation protocol. So I'll start by telling you about just us kind of looking at what our plants are doing kind of in our natural systems. So again, huge distribution, right? And even within Chatham County, if you look at the Chatham County map, there's a lot of variation of where marshes are located within our coastal system, right? Some of them are located really close to the ocean. Some of them are located further upstream, right? Or more inland. We would expect maybe that the marine influence is more in one than the other, right? They vary in anthropogenic disturbance, right? They vary in a lot of things that I can't measure at this time, but there's a lot of variation within our county system, right? And then, of course, climate varies from year to year. So we were really interested to see if we could see patterns either spatially, like within the county, and also temporally, which would be from year to year. And so our purpose was to characterize those patterns. And the reason we wanted to do this is we wanted to be able to say, okay, here in coastal Georgia, here's the best time to collect seeds, and here's the best marsh to choose, right? To kind of limit and help guide where to have these seeds collected. Again, currently not legal to collect seeds in the marsh, okay? You have to have a permit. So to do this, we selected 3 marshes of varying marine influence in Chatham County. We chose Lazaretto Creek Boat Ramp. We chose places that were easily accessible, right? I don't have a boat. So, and let me tell you, from getting stuck in the mud, there's only so many places you can go in the marsh before you are really putting yourself in danger of fire rescue, right? Helicopter removing you from the marsh. So we did Lazaretto Creek Boat Ramp. This is our location with greatest marine influence. Kind of in the middle is Rodney Hall Boat Ramp. And then we did Halcyon Bluff Community House. That's my neighborhood. So I live in Halcyon Bluff, and we have a community house. And so I just kind of went right off the marsh there. We visited them every 2 weeks between October 1st and November 31st. Every year from 2019 to 2022, and we would collect seeds from all the marsh sites every 2 weeks when they were available. Just to show you kind of what these sites look like, here is Lazaretto Creek. We don't have the equipment to continuously monitor salinity, but we measured salinity every time we went out there. And over that whole time period, it ranged from about 25 to 26. So this was our one closest to the marine influence. Rodney Hall boat ramp was kind of in the middle. You can see these two are public areas. Lazaretto Creek has a single, double or 3-lane boat ramp and a parking lot. It's right off the road. Rodney Hall is Butterbean Beach if you guys need a call from something else. You can see this one has 2 double-lane boat ramps. It has a really large parking lot. You can see there's a kayak group that kayaks from here. There's a beach over here that people play. My kid has drawn many times in the mud. And then there's like a big picnic shelter. So both of these are publicly accessible, right? So we would expect some anthropogenic disturbance. This is where my marsh, I call it my marsh, just letting you guys know. This is where the marsh in my neighborhood is. It's Halcyon Bluff. You can see it's kind of on the corner here of the marsh, and we were kind of all in this area here. This is kind of less disturbed because it is in the Vernon River, which is a whole nother story, but the Vernon River is not doing so well, right? But it's less disturbed by things like boat traffic and human pressure, like physical pressure walking in the marsh. So there were 2 kind of aspects of reproduction that we looked at. One thing we were interested in, I'm going to call phenology. So that's kind of their reproductive events. So when are they producing flowers? When are they producing fruits? When are they producing seeds, right? How many of them are producing seeds? And so to do this, we only did this in 2020 and 2022 at Rodney Hall boat ramp, and in 2022 at the Halcyon Bluff Marsh. And what we would do is we would establish transects through the marsh. And so if you're not familiar with transects, these are 2 of my students, and you can see this kind of rope coming out here. So it kind of looks like, you've seen the big meter tapes. So we just take one of those, we start in the marsh, and we just walk back in a straight line to about 20 meters, and then we stop, okay? And we plonk it down. And then what we would do is we would take quadrats, which are these little plastic squares that were about 0.5 meters squared. And every 2 meters, starting at 0, we'd plop it down, right? And then we would assess the plants. We would count the number of stems. We would count how many of them were flowering, how many of them were fruiting, and how many of them were seeding. And then we did this every 2 weeks, right? During that time frame. We actually started this in September, but let me just state that like Spartina starts flowering in July, so we definitely missed a good chunk of the reproductive season. But, you know, it is what it is. To collect seeds, we would kind of look closely and see if the seeds were ready to collect. We could figure it out because they call it a spikelet, the thing that all the seeds are on. And if the spikelet was like completely brown, we would try by putting a plastic bag over it, and then if we barely touched it and the seeds came off, we would collect them. So we would collect somewhere around 20 to 30 of them. We would bring them back to the lab. Once we were back in the lab, my students would count the number. They would remove the seeds, count the number of seeds that were on each spikelet, and then put them in water and in cold conditions, because the plant had to go through what we call cold stratification, where it spends at least 2 months in cold, wet conditions to break dormancy so it'll germinate. And that was based on other propagation protocols. Conveniently, that was Christmas break, so it worked really well on an academic schedule. So when we came back in the spring semester, the first thing we did was tested to see if the seeds were viable. This means, was there an actively growing embryo in the seeds? And to do this, we use something called the tetrazolium test. It uses a dye called tetrazolium chloride, and it stains for metabolic activity. So if you have an embryo in there metabolizing, right, using energy and growing, it'll stain red, right? And so this is an example where this is a seed that does not have an actively developing embryo, and this one does. Okay, so that just tells us whether or not the seed has a plant growing in it. Now the downside is it kills the seeds. So we only did it on 25% of our seeds. When we figured out with spikelets with seed viability greater than 10%, we then placed in culture dishes with water, and we monitored their germination for 14 to 21 days. Again, putting them in culture dishes like this was something that I began based on a protocol. So I worked in Mississippi for a couple years with a professor who was growing Mississippi native Spartina, and I used his protocol, right? So a lot of this started from just kind of based on someone else's work. It makes sense though, because during this season, they're kind of in the mud, they're floating around. So they would be in wet conditions. And as a matter of fact, according to literature, if you let Spartina seeds dry out, they're dead. So they have to stay wet. Okay. Lots of graphs. I'm sorry. I will try to explain what we're seeing here. So first, let's look at, and I just want to toot my own horn and say this was currently in press at the Journal of Coastal Research. And thank you. So this will be out in publication soon. So when we look at flowering, okay, so what you're looking at is this graph is showing the percent of individuals flowering, the percent of individuals fruiting, and the percent of individuals seeding at different collection periods. So from mid-September to late October, okay? And this is the black bars here are 2020, and the gray bars are 2022. So this is us looking at that single site if there's a difference among years, okay? And what we found is overall, there was more reproduction in 2020 than 2022. In 2020, 25% of individuals reproduced. And in 2022, it was like 17%, right? So only about a quarter of individuals are reproducing within a given season. We also saw just kind of a shift in the season. And so they started seeding earlier in 2020. So seeding a little earlier in 2020, shown here in black, and then a little later in 2022. But they started flowering later in 2022 than in 2020. So we're seeing that there are some shifts kind of from year to year in when things are happening, right? Does that make sense? Cool. When we look at sites, right? So that's this side. So remember, we looked at 2 sites in 2022. We looked at Butterbean and we looked at my marsh, right? Marsh by my neighborhood, just to see if there's any difference within the coastal system. And so we didn't really see a whole lot of differences. What we really just saw was the peak of reproduction, like when most individuals were in like some form of reproducing, was different among the sites. But overall, we saw some similar trends in all of this data. We saw that flowering starts to go way down after sometime in late October or late September. So you can see that our flowering decreases on both graphs. So flowering goes down, and we see seeding generally increases over that growing season. And we see that the most seeds are being produced somewhere in mid-October to October, early November. So we do see there's kind of this key period time when it's best to go get seeds, right? With the reproduction, sorry, science is more graphs, right? But I tried to kind of make this easy to see. So we have number of seeds per spikelet. This is how many seeds was actually on that seeding stalk. The proportion of viable seeds, how many of them had an active embryo, and the germination rate, how many actually germinated. On each group here is a different site, and then each bar is a different year. So starting with 2019, 2020, 2021, and 2022. And yet, they're all over the place. So hopefully the first thing you can kind of see is there wasn't like a really clear trend where we're like, well, that's obviously more than the other. But we do see some trends if we look a little closer. If we look kind of at, and these letters, guys, are statistical significance that I have to put in publications. We run statistical tests to make sure our data are really different, right? And then we report that. So I took a lot of that out, but the letters are still here. And the way you interpret that is if two groups share a letter, they're the same. And if they don't share a letter, they're different, right? And so if we look at sites, right, we can see that this trunk here with number of seeds per spikelet is bigger than these two, right? And so we obviously saw that one site produced a lot more seeds. Ting, ting, ting. All right. So, we can also see that with the seed viability, we can also see with spatially, we can see this chunk here, which was that lacerated creek, has more viable seeds than this chunk here, right? So Butterbean didn't have a whole lot of embryos compared to the other two sites. Germination rate didn't matter, right? So we didn't see any spatial patterns. With our annual patterns, this was kind of all over the place. We didn't really see any difference among years for number of seeds per spikelet and germination rate. So the year was not really affecting how many seeds they made and how many eventually became seedlings, but it did kind of have an effect on how many embryos were actually developing. I will say that our viability is much greater than our germination. So we found that the potential to make babies and produce seedlings was a lot higher than what we actually got. And that's pretty standard for plants, right? So, you can have plants, germination is a safe site. And so they really have to have like these perfect set of conditions in order to germinate. So that's pretty normal to have more potential than what you actually get out. Okay. Does that make sense? Kind of like you guys have planted seeds, right? You put a bunch of seeds in and not all of them do usually germinate, right? We got some unexpected results. Again, science, right? But we basically found that we weren't only seeing variation within these sites. This was actually a surprising site. We found that the reproductive effort within populations was a lot different. And so what this graph is basically showing is this is the range of seedlings produced by different sites in different years. And they range from 0, meaning that those seeds did not germinate at all, to like 66 and 21 and 29. What these two numbers are the median and the mode. So that's really representing what's the most common value I got, right? When I looked at that data, what was the most common value? So what was most commonly the number of seeds that germinated and became seedlings with each of my plants? And a lot of them gave me nothing, right? And so we kind of found that most individuals produce seeds that maybe like 1 or 5 of them germinated, right? But not a whole lot. 'Cause I mean, there were like hundreds of seeds on these guys. But then we had a couple individuals that were like 66 and like 50 and like 30, and they were very few. And we named these super seeders, right? So we have few individuals that are just gang-busted and making babies. And they're contributing significantly to the population structure, right? Why are they doing this? We have no idea, right? So what influences an individual to become a super seeder? We don't know. Right? We did a very really short, quick experiment with an honor student where we collected spikelets with seeds. We actually collected soil and we measured some plant characteristics because we wanted to see if we could link the plant characteristics, like do the taller plants have more reproduction or is it a bigger stem? We looked at the health of the plant. We looked at like salinity and nutrients. We looked at all these things to see if that was influencing them. And basically, I don't think you can really see these figures very well. But what we ended up with was when we plotted like all these things that we measured against like the number of seeds and seed viability and germination, we still ended up with our super seeders, right? So most things fell within the cloud. But then the ones that were out of the cloud that made lots of seeds were still lined up with the stem diet. So basically what we found is like, nope, soil characteristics and plant characteristics, nothing mattered, right? We don't know if it was because we only took a snapshot. Like, do we need to go out and measure these things like routinely, like every month during the growing season? Does it need to be more common? We don't know if it's genetic, right? We don't know if it's some microbes that are associated with the roots. We don't know. So this is kind of this open area that I would love to explore further once I can make all the collaborations and get the money to buy all the stuff I need, right? Science takes money. So, all in all, what we found is that there are year-to-year variations in when things flower. There are year-to-year variations in whether or not the embryos are viable. And we think this probably is due to annual variations in climate change or climate. We found that the 2 years with like the lowest viability and the lowest viability was also linked to years where we had lower precipitation and longer, hotter summers, right? So we think that might have had something to do with it. We also found seasonal variations, right? So we found that there were certain times of the year or certain times of the growing season when there were a lot of seeds. There were certain times when you had a lot of seeds filled with very active growing babies, right? And we basically found that this is kind of showing our collection periods, but your biggest bang is to gather things later in the season, right? So not only are the number of seeds increasing, but the viability of the seeds is also greater later in the season. And this could be because early in the season, you got little guys flowering, just a few, but then kind of in the middle of the season, you got everyone reproducing. So there's a lot of pollen being exchanged, which increases seed production. So we established kind of this graphic. This is in the propagation protocol that kind of shows when things are happening with Spartina in Georgia. They're probably flowering in July, but they kind of stop somewhere around late September, early October. They're kind of fruiting throughout most of the season, but the solid lines where you're getting maximums. So you get maximum seed production here later in the season. And this allows us to recommend that if you're going to collect seeds for nursery production, to collect those seeds later in the reproductive season, right? So, and we've started now, like my lab, we don't go out till mid-October now because it's just, what's the point? So, we save our manpower for that. With our spatial patterns, we did kind of see the marsh by my house, which was the furthest from the marine influence, tend to have greater number of seeds per spikelet. And they have pretty moderate seed viability, meaning that might be the best location of the ones I had to kind of look at, to kind of collect at. I think this might be a result of stress tolerance. Salinity is something that, Spartina is not taking salty water and being like, I love salty water, but they're actually using their energy to do processes and metabolic chemical processes to like take that salt and shunt it here and send it over here. And they basically, it's kind of cool. They have these little salt glands and they'll kind of concentrate the salt somewhere and send it up the plant, and then it'll come out of those salt glands. So if you're ever in the marsh and it hasn't rained for a while and you run your finger along the blade, you'll get a bunch of salt, right? And then it rains and it washes the salt off. That takes energy, right? Which means that energy that they're using for that response to salinity, they can't use for reproduction. So it could be that, we call it allocation. It could be that their surplus energy that they use for maintenance and reproduction is being shunted over to stress tolerance. And that means that the closer to the ocean where the salinities may be higher, they're just not able to reproduce at the same extent. We also found that Rodney Hall, the seeds there were kind of like our less productive plants. And this could be due to anthropogenic disturbance. Both of those are really large boat ramps with a lot of boat energy that could be disturbing our plants, right? And again, they're stressed, so they're not reproducing. And so our recommendation is to try to get further into that system, right? Where you're a little further away from that marine influence. I am not going to get through all of this. So, we are going to power ahead. We'll skip this, even though it's very interesting. You guys can look at the book, the protocol, if you're interested in seeing how I did that. Anyway, I do want to tell you some of the things we've done to just look at alternative nursery production. Wow, I am really running out of time. Sorry. Get me talking. So basically we wanted to look at other ways that you could maintain the plants once they're grown. And so, like I mentioned, traditional nursery production just means they put them in these sub-irrigated trays. They add like Miracle-Gro. And that's, again, not very similar to what they're going to experience in the marsh. So we wanted to look at if there were ways that you could produce them in the greenhouse that would help mimic salt conditions, these marsh conditions, and better prepare them. One thing we did was to look at growing them in constructed wetlands. And so we were more interested in wetlands that were used to remediate effluent from aquaculture production, which is fish farming. So we were interested to see, especially for marine aquaculture, if this was an option, where instead of having to do water changes, you can basically set up an aquaponic system and produce salt marsh plants, which are used to salinity, so that the salt water can be cycled back into fish production. And Spartina is great for this because it is able to grow in freshwater and saltwater, and it can tolerate low soil oxygen, so it's fine with being in that constant flooded conditions. So what we did is we wanted to see if we could produce them in a constructed wetland designed for aquaculture. And so this was a master's student that worked on this. And we basically wanted to see, is there any difference in growth between growing them in a wetland versus growing them traditionally, and do they actually help aquaculture effluent? So we built a wetland out back. So it is not this pretty anymore. I think my students can attest that this is not what my marsh looks like. But we built this wetland out back. We were able to basically we had these different cells, and the water was constantly cycling to a tank here where we were doing fresh water because we didn't have a cover, so we couldn't control salinity. And we were using tilapia. And so we basically had 4 different cells. We had 2 cells that we stocked with Spartina, and then we had 2 cells that we left empty. And we measured water quality parameters from all 4 cells and the tank on a weekly basis. And then on top of that, we had 2 tables that were exactly the same size as our wetland cells, where we grew the same number of plants in traditional methods. So we fertilized monthly with Miracle-Gro, and we kept them sub-irrigated. And we measured plant growth and productivity for like 12 weeks. And basically what we found was that in terms of our growth metrics, we measured growth rate, how many stems they grew, how big the stems got, how tall the plants got, how much biomass they made. And what we found is that there was no difference between our traditionally grown plants and our aquaculture grown plants. So that was really great. We did have some plants that we propagated from cuttings from the marsh, and those grew faster than our seedlings, which makes sense because they're older, right? So they have stored energy that they can put into growth where our seedlings are just getting a start. But we basically also found that the water quality parameters didn't really matter. It didn't differ, and overall the system maintained itself in like this healthy water quality for an aquaponic system. So that told us that Spartina can be grown in these above ground wetlands, and that it can be used to remediate aquaculture effluent. So this is a potential. Now we overwinter and just store my extra Spartina in the wetlands. We don't have it set up to a fish tank, but we keep it aerated. And what's really nice is it became like a little habitat for like a little hotspot on the Armstrong campus. These are all frog eggs. So we had tons of frogs. We had grasshoppers. We had these really cool slugs. We had piping plovers that were hanging out and feeding around, and they stuck around for a couple more years. And we would come out, and you could hear, I don't know if you guys can hear it, just be frogs singing all day long. So it was really cool. It ended up being a little bit of both. I'm going to stop there because I obviously talk too much. But I just have so much cool stuff. But I want to acknowledge, I have done some work in collaboration with Joel Koska at Georgia Tech. I didn't present it to you, but we did some work looking at how adding salt marsh soil to nursery production in the greenhouse influences plant growth by bringing in those native soil microbes. And he's a microbiologist and found that we pretty much established a rhizospheric bacteria community, like a microbial community around our greenhouse plants just by adding some salt marsh soil that is very similar to what they see in the marsh, which may help with transplants. So we're doing further research with what he likes to call probiotics for plants, where you can isolate these rhizospheric bacteria, spray them on the plants, and then transplant them in to help them get a good start. I want to thank the University of Southern Mississippi Center for Plant Restoration. That was where I worked, and I based a lot of the beginning things that I did on the protocol for Mississippi. But what I've done with my protocol is really extended that and tried to specialize that for Georgia. I've had so many undergrads work on this in the past 7 years. I work with a lot of undergrads, usually somewhere between 6 to 9 a semester. So there's been a lot of students that have been involved. I had 3 graduate students that have been working on this. And then I've had funding through the Georgia DNR Coastal Incentive Grant for 3 years. So 2 different projects. And then the Georgia Southern College Science and Math Research Grant supported one of my grad student projects. So it's too much. All right. So this QRL, if you're interested in some of the other things that we're doing, or this QR code, will take you to my website. So my lab has a website. And on that website, we have our propagation protocol as well as a very low tech demonstration of some of our techniques in growing Spartina. And I'm sorry I had so much to talk about, but thank y'all, and I'm happy to answer questions or talk further. Look down here. Yeah.
Did you say, I think I understood correctly, the output at the public boat ramps or the performance of the grass was a little
lower
than the private?
Yeah.
One, do you think that would change if you went across the bank or down the bank, like further away directly from where the boats are putting in the recreational traffic is?
Possibly, right? There's so much we didn't measure, right? But basically what boat wake does is marshes establish in low-lying coastlines that have like a certain amount of wave energy. And here on the Georgia coast, we have very little wave energy, which is why we have so much expanses. And when the wave energy increases, it starts to erode. And so those boat wakes, the wake that comes off is oftentimes at a higher speed than the natural wave energy. And so it erodes the banks. And then there's pollution, right? Oils, biofoulants, right? Things, anti-foulants to kill like anything to get on your boat. And so we hypothesize that that has an impact on reproduction from kind of the stress kind of aspect, but without being able to measure like everything in the marsh, right? Like I couldn't measure salinity or nutrient availability or tidal amplitude or, you know, all of that stuff. I can't say 100%, but I think it might be better if you went further down. Unfortunately, my marsh, there was a hydrological event and a bunch of dead stems smothered it. So it's not, it's not there anymore. Yeah.
Is that what you were saying about the Vernon not doing?
Oh, well, it's also kind of polluted. So yeah.
Recently, you guys got more polluted, or?
We had them come out and test like right around our area, because it wasn't great. Yeah. That's probably something for a river keeper to come talk about.
That was pretty far up there, though, right?
No, they came out to our dock.
Yeah, but your dock's pretty far up there, right? I don't know, just judging by the picture.
I'm on the south side.
Oh.
Yeah. So, yeah, I mean, not too far. Yeah.
If you were looking for a living shoreline project, where would you find your marsh if you're saying there aren't nurseries that have it available?
So most of them, so they have a variety of different ways of going about it. I know that there is a native plant nursery who will bring plants in for marine contractors that do the living shorelines from some nurseries in South Carolina. And so a lot of them are coming from South Carolina. And then some of them will actually try to dig them out to restore the shoreline and then put them back in. So maintaining the local. But sometimes it depends on the shoreline. Like if they're already there, you can try to salvage them and then replant them. But if they're not already there, I think they're mostly coming from South Carolina. Yeah.
And given the reproductive cycle, what's the most productive time to plant, to reestablish?
So, my suggestion, unfortunately, I haven't been able to actually translate anything into the marsh quite yet. It's been permitting issues. But my recommendation was that they resprout, like basically in the fall. You know how the marsh turns brown, right? So basically when they are allocating all of their energy to reproduction, they're also bringing some, well, not all of it, they're also bringing some to store for below ground. So they stop assimilating it into growth. So around fall, they're starting to move everything into storage organs to go to sleep for the winter. And so my advice would be like spring, right? Spring, kind of this time of year would probably be an optimum time to bring seedlings in because they're gearing up, because their big growing season is the summer. So they're gearing up to get started. Yeah.
Have you done anything besides Spartina as far as the other plants that support a living shoreline?
So I have fiddled around with sea oxeye, which is that shrub that has kind of like the aster flower. My focus has been on Spartina just because, you know, that's where my funding source is. And once you get a grant, you gotta follow the funding. But I haven't really explored it a lot here. So, really I've been mostly focused on Spartina. I used to work on coastal sand dunes and I grew some pennywort, but no one likes pennywort. So, so I won't tell you about that. But yeah, I know that Jekyll Island is currently, I think they're partnering with the Georgia Native Plant Network or something like that. Huh? Well, no, they're doing native. They got a new greenhouse and they're getting into native plant production. Yeah. So I know that they're branching out there and we're trying to collaborate. But right now with just my limitations, and I only have like 4 benches in a greenhouse. So I'm kind of limited on what I can do. Yeah.
You said you collected seeds going into the marsh on that transect. I was wondering if the ones that were deeper in produced more seeds because they would be away from as much wave action.
So
trampling in the mud and everything else.
Well, that was mostly just for the reproductive stems. So when we went out, I had a great picture here of my child just like running out of the marsh. She used to come help us. But when we kind of collected seeds, that was for like the reproductive like status. But when we collect seeds, we kind of just wander around and find some that are mature and clip them. So we haven't really tracked on where we collect them from. However, there are 2 what we call growth forms of Spartina. So you can probably notice this next time you're out in the marsh, but along the creek beds, they're super tall, right? But then when you get back a little further, they're really short. So it's a tall form, very, very cleverly named, the tall form and the short form. And they're actually related to how often that area is getting flushed by tides. And so the tall form is growing along the tidal creek where it's constantly getting flushed. And that flushing brings in nutrients, it oxygenates the soil, it removes some of the built up salts, right? And so their stress is a little less, right? Whereas as you get further back, that's only getting flooded in like super high tides. And so the soil, even though it's not getting tidal flooding, it's still really close to the water table. So the soil's all like full of water, there's no oxygen, and the salt evaporates, and so it's really salty. And that's where you find the short form. And so they think it could be a combo of genetic and due to those different stressors. And so one of the things that we're exploring right now, I'm getting to a point, is we're actually seeing if those 2 forms differ in reproduction. So does one produce more seeds? Does one germinate more, right? And so we got some results from this year, but we need to repeat it over time. We did see a difference in viability in forms and germination. So it could be that the forms differ, but that's something no one's really looked into. And so we just started with that. We're also going to track them in the greenhouse because they've never been in the marsh, right? So the seedlings that we produce have never been, they're maternal, their moms were, but they've never been in the marsh. They're grown in potting soil. I have some over here if you guys want to take a look at them. We literally germinate the seeds, right? We literally germinate the seeds just like you would at any plant nursery. We germinate the seeds and we put them in a little peat pellet and then we put them in potting soil, right? So we are actually going to track the seedlings we grow from the different forms to see if they maintain those differences when they've never been in a marsh, right? So we're hoping to contribute to the discussion of is it genetic or is it environment? Nature or nurture, right? Yeah. Yeah.
I have a question going back to the forms.
Yeah.
Spartina. And so, from the way I understand it, there's like high marsh and low marsh, like the marsh that's always flooded and the marsh that's getting more cyclically flooded.
Mm-hmm.
So, if you're propagating from one type or another and you're putting it into another project, how important do you think it is to make sure that?
That's a question we're exploring, right? So, I mean, that's a great question, right? I mean, there's a recent study that found that they basically think that the Spartina plants vary in their tolerance to salinity and their tolerance to, just like we vary, right? We're all different, right? So they think that they vary in kind of how well they can tolerate salinity and how well they can tolerate low oxygen. And that kind of determines where they kind of occupy, right? And so there is that question, like or even tidal inundation, like how well they can handle being underwater. And so if we collect from the high marsh and then we plant in the low marsh, are we bringing in something that's maladapted, right? That's one of the things we're kind of looking at, like, if we grow them without any marsh, are they going to be the same, right? So that might suggest they might be able to be plastic enough to move into that marsh. But that's a good question because honestly, the high marsh or the low marsh is super hard to collect in, right? That tall form is hard. Guys, I can't tell you how many times I've been stuck in the mud, right? It's hard. And so, without like a boat and, stuff like that, it's hard to get to those areas. Yeah.
In terms of adaptation, I think you were saying that you had two groups that you looked at in terms of the Spartina growth. One was aquaculture in freshwater, and one was traditional methods. But since the Spartina lives in a pretty saline environment, it seems to me that third group, even though I understand why you couldn't do it.
I did do it. It's just, I did do it. I didn't show it to you.
Okay. Tell us the result. That would be important because otherwise you may be selecting for plants that aren't going to like the saline environment. Right.
So that's a really good point. And so we actually looked at that. And that was, if I had 3 hours to talk to you guys, we could have covered it. But we did an experiment where we grew them in fresh water, and then we grew them in 13, salinity of 13, right? Saline water, and we compare growth, right? And what we found is that they prefer fresh water. We also did an experiment where we tried to overwinter seeds and germinate seeds in a variety of different salinities at like 0, 10, 20, and 30. And they preferred fresh water and preferred to germinate in fresh or slightly saline waters. So there is a preference in this plant for fresh water. And I think what the reason why we find it where we find it, right? And you don't have it growing back in fresher areas. Spartina sucks at competing, right? It's really bad at competing with other plants for resources. So it occupies an area in the marsh where it's so stressful that only it can tolerate those stresses. So once those stresses are ameliorated enough for other plant species to grow, they quickly outcompete Spartina. And so this is something that is another avenue that my PhD student is moving into, is because mangroves are migrating north, right? And they occupy kind of the same niche as Spartina, right? And so there's this kind of fear that mangroves are going to eventually replace Spartina, right? Which would totally change our marshes. And so one of the avenues that I'm interested in going in with her is to look at Spartina mangrove interactions, right? So start with no marsh conditions, see how they interact with each other, then move into kind of simulating marsh conditions. I've built marsh mesocosms, which are little marshes and tubs, and then maybe moving it out into the marsh to see if that's really what's going to happen, right? Because who knows, mangroves might not like 10 feet of water. You don't really know. So yeah, so it's really, that's what's kind of, and you can go out in the marsh and you can see like almost clear lines where Spartina is and then where the other vegetation is. And that's because it cannot compete. Yeah.
Well, this has been wonderful and informative. So thank you so much for sharing.